course, no symposium on software tools would be complete without WGCNA, and Peter Langfelder from the Horvath group is going to tell us about this this tool that probably almost all of us have used, or at least hope to use at some point.
All right, thank you. So I'm afraid that my talk will be on something that's much older, and so I'm just going to present a very brief overview of how weighted gene co-expression networks or weighted correlation networks in general can give you some biological insights. And the tagline is from 1 million correlations. And in fact, in modern analyses, it's more like 1 billion correlations. So WGCNA, again, this is going to be a very brief overview, is a compendium of methods to analyze what is known as high-dimensional data. So for me, that means thousands up to perhaps millions of variables, which could be gene expression, methylation, proteomic, fMRI, metabolomics, and other data that are measured across multiple samples. And I want to emphasize that you need at least 20 samples. More is better. The aims are fairly down-to-earth biological. It's to identify candidate genes for interventions, formulate new hypotheses, gain biological insights into pathways that may be associated with the condition that you're studying. You can also, if you have several data sets, study commonalities and differences in the organization of the high-throughput data that you study, so be it transcriptional organization or methylation or proteomic, between conditions, between species, between tissues, between data sets from different laboratories, which can sometimes be very different, and so on. So very briefly, what do you need to carry out basic WGCNA analysis? So you need the high-throughput data set. In basic analysis, you just need one. In an advanced analysis, you could use several independent data sets. And you need a sample trait or traits. So it could be a disease versus control indicator. It could be a genotype. It could be a quantitative trait like body weight or proportion of fat and so on. So the traits could be numeric, ordinal, or binary. It could be pretty much anything. In terms of implementation, WGCNA is currently implemented as a package for R. So the bad news is that if you don't know R, you have to learn it. The good news is that once you learn R, it'll help you a lot in your research in the future as well. You don't have to start in the dark. We have a lot of example scripts and tutorials. If you Google search or whatever your favorite search engine is, for WGCNA tutorials, the first hit should take you to the webpage with tutorials. I also listed the webpage here, but I don't think you'll be able to remember it. Certainly searching for WGCNA tutorials is much easier. So just a very brief overview of the main steps of a basic WGCNA. The first very important step I want to emphasize is because a lot of times people don't take time to do this step properly, and that just sort of sets them up for a lot of trouble further down the line. You have to ask good questions, and you have to prepare your data. So you have to make sure that you have enough samples, that the design is appropriate, that you control for batch and other technical effects. Pre-processing is quite crucial, so WGCNA really needs decent data, otherwise it will not give you decent results. Then the first step is to construct network and identify modules, and there's a little bit of a cartoon there. I wonder if, I guess there's no pointer here. Or is there? Oh, there is. Okay, perfect. So the modules are identified by clustering, and so you get these blocks of genes that are co-expressed or variables that are correlated together. And the idea is to reduce the thousands, tens of thousands, or hundreds of thousands of variables into a relatively small number of modules. Oh, should I use the microphone actually?
Well, you can just stay close to it.
Oh, I see. Okay. You know what, let me take it. I like to stand a little further. Once you have the modules, then you identify biologically interesting modules. And that could be done in two ways. You can relate the modules to a sample trait. So for example, here, this is again just a cartoon, but here the trait is on the x-axis. It's a numeric variable, and some of the modules sort of stay flat, so they don't change very much, and some of the modules change strongly. So those ones that change strongly, those would be the ones that are more interesting. You can also annotate the modules, because those are groups of genes, using functional enrichment. So it's easy to do on genes. It's a little more challenging to do, say, on the methylation probes, but there are tools that can do it very well. And then you identify key genes and interesting modules, and the hypothesis that underlies network analysis in general is that hub genes, so the genes that connect to a lot of other genes in this little plot here, are often the most important, biologically important genes or variables in the module. So that's a very brief overview of the main steps of WGCNA, of a basic WGCNA analysis. Now, oftentimes people have several independent data sets that sort of measure the same or very similar things, or they are comparable. And so again, after you've asked the right questions and you prepared several of the data sets, you can carry out what's known as a consensus WGCNA. And the aim is to find common modules across a few independent data sets. So in this cartoon, there are 3 different data sets. And the idea of the consensus analysis is that you create networks in each of the 3 sets, and then you combine them into a consensus network. And roughly speaking, the consensus is a network that all 3 data sets can agree on. So for 2 variables to be connected strongly in a consensus network, they have to be strongly connected in each of the 3 input data sets, input networks. Now, once you have those modules, the rest of the annotation and finding key genes and so on is very similar to the basic WGCNA analysis. So what are the advantages of consensus network analysis? First of all, because you are looking at 3 independent data sets and you're taking sort of their consensus, if you find a module, it means the module is present automatically in 3 different data sets. So you have a double validation already right there. That's usually quite valuable. But apart from that, the commonalities and differences in how the modules relate to sample traits are also often interesting. So this is a plot that you have seen before, but now I'm going to reinterpret it in this example. This is one module in 3 different data sets. So the 3 different data sets correspond to 3 different ages here in this analysis: 2 months, 6 months, and 10 months. Again, the Q, just think of it as a numeric variable. And you can see that in one of the data sets, the module just doesn't really vary very much with the numeric variable, with the condition, I suppose. Whereas at the other 2, in the other 2 data sets, the module changes very strongly. So there's a clear difference between the first data set and the other 2 data sets. So if you know what the data sets were, in this case, one was from young mice and the other one was from adult mice or older mice, something is changing in the older mice. The older mice do respond to this quantitative variable, whereas the young mice does not, or not really. So this is something that a consensus analysis can give you. You can, of course, figure this out for individual genes, but if you have 20,000 genes, it's kind of hard to interpret, whereas if you only have 10 or 15 or 20 modules, it's much easier. And the module analysis can tell you, okay, this module is enriched in neuron markers, and it's enriched in GO-term cell periphery. So there is something going on with neurons that at 2 months, nothing changes, but at 6 and 10 months, they start, the expression of those genes starts to go down. One feature of consensus analysis is that all the data sets are treated on the same footing. So you don't have to prioritize. I'm going to define a module in one data set and then see how it behaves in the other data sets. No, you define a module across all three data sets at the same time. There's a complementary question that people often ask, which is that sometimes you already have an interesting module from your analysis, and then you submit the article, and then the reviewer says, oh, but there's another data set that's very interesting, and if you don't find that module in the other data sets, I'm not going to let your paper be published in this journal. Sometimes it happens. So fortunately, so basically, more generally, the question could be, is the module condition specific, or could it be also found in data sets from other conditions? Could it be validated in data sets that measure the same thing but are from a different laboratory, are measured on a different platform? One is a microarray, one is RNA-seq, and so on. The question is somewhat complementary to consensus analysis. So here we're not treating the data sets on the same footing. There is a clear reference data set in which the module was found, and perhaps multiple test data sets in which we just want to know whether the module is in some sense preserved. Are the genes or the variables correlated in that other data set? So naively you would say, well, okay, so let me do a full analysis of the other data set, find module, and see if there's overlap. But there's a lot of things that go into module identification, a lot of variables, and people might say, well, you haven't found it, but if you used another clustering technique, you would find it, and so on. Fortunately, network analysis can free you from all these constraints. So the network analysis is done in the reference set, but in the test set, you don't need to do a full module analysis. You just construct a network, and basically you see if the modules look like real modules. So again, I'm not going to go into the details. If you want to go through with this analysis, we have, again, examples, tutorials on our webpage. So here are resources. If you want to run WGCNA, I suggest that you first visit the WGCNA webpage. Here's the URL, but again, if you search for WGCNA, it's much faster than typing it in. There are tutorials, 3 example analyses tutorials. If you have never done WGCNA, you can get started. There is another website, the Horvath Lab Co-Expression Network page, where Steve has put a lot of links to articles where our group has used WGCNA and other approaches for analysis of data. So there's a lot of other examples that are perhaps not as pedagogical, but they could be more informative if you have a certain specific question in mind or a certain specific application in mind. There's also a book that Steve Horvath wrote, Weighted Network Analysis, Applications in Genomics and Systems Biology. As you know, books are often quite expensive. The good news is that for UCLA, students, faculty, staff, the book is free. So if you go to the Springer website and you are logging in from a UCLA computer, or you are connected to a VPN to UCLA, to the UCLA network, you will see this. You have access to this product online. Click here, and it's free. So very much recommended. I just want to mention a few limitations of network analysis that are summarized basically by this picture, saying that network is not a magic wand and a network analyst is not a magician. As all computational analyses, WGCNA requires that you put in well-designed data and you have a question that the network analysis can answer. I don't want to sound sort of disparaging, but you know the old garbage in, garbage out still applies, even for WGCNA. So plus WGCNA also puts in certain additional assumptions. For example, that the network analysis and the assumptions that network analysis makes are actually appropriate to your problem. So as I mentioned, network analysis assumes that highly connected nodes in the network, in the module network or in the full network, are actually the ones that are most important. And this is sometimes not true. So in that case, network analysis will not help you in your analysis. So I think I'll stop there and I'll take questions.